Welcome to another deep dive,
We're tackling a disease you might not have heard of.
but it affects millions globally.
Okay.
Schistosomiasis.
Yeah, it's one of those that really kind of flies under the radar.
Big burden in many parts of the world, though.
Right. And we are diving into a scientific review from March 2024.
That really digs into the challenges of diagnosing and monitoring this disease.
Mm-hmm.
And get this.
It explores how cutting-edge science could change the game entirely.
Yeah, it's exciting research for sure.
Okay, so for anyone who's new to this, let's start with the basics.
What exactly is schistosomiasis?
Well, in simple terms, it's an infection.
Caused by parasitic worms,
Okay.
specifically a type called Schistosoma.
And these worms have a life cycle that's dependent on both humans and freshwater snails.
So it's not just a human problem then.
It's an ecosystem problem, too. Exactly. It starts when infected snails release microscopic larvae into the water.
Okay.
And if someone comes into contact with that water, say while swimming or washing clothes,
These larvae can burrow right into their skin.
ugh, that's pretty unsettling.
And I'm guessing it doesn't end there.
Unfortunately, no.
It does not end there. The larva matures inside the human body.
Okay.
They reproduce and start producing eggs.
Some eggs pass out of the body through urine or feces, but others get trapped in tissues like the liver or bladder.
And those trapped eggs are where the real trouble starts, right?
Yes, that's right.
They trigger the body's immune system,
Okay.
leading to inflammation and, over time, serious damage to the organs.
We're talking about things like organ enlargement, problems with growth and development,
Mhm.
and in severe cases, it can even be fatal.
Okay, so definitely not something to mess around with.
But this is where our scientific paper comes in.
Right.
It points out some serious flaws in the way we currently diagnose and monitor schistosomiasis.
It does.
Isn't it?
The standard method for diagnosis involves examining stool or urine samples
Okay.
under a microscope to look for the parasite's eggs.
It's a technique called CATO-CATS. It works, but it's not perfect.
Imagine trying to find a few specific grains of sand on a beach.
Right.
That's how challenging it can be to spot those eggs, especially if the infection is mild.
And missing those mild cases is a big deal there, right?
Absolutely it is.
It's becoming clear that even low-level infections can have long-term health consequences.
If they're not caught and treated.
Mm-hmm.
Plus, these milder infections are becoming more common.
Likely because mass drug administration programs have been so effective.
In tackling the more severe cases.
So we've knocked down the worst of it.
But now we need tools that are sensitive enough to catch those low-level infections before they become a problem.
Right.
Hmm.
Makes sense.
But diagnosing schistosomiasis is only half the battle, isn't it?
Mm-hmm.
We also need to figure out
How much damage?
those sneaky parasites they're
Yeah,
causing.
You're right.
Traditionally, we've relied on the number of eggs detected to gauge the severity of the disease.
However,
Recent research shows that this isn't always a reliable indicator. So you could have a low egg count and still have significant organ damage.
or vice versa.
That's precisely the issue.
The gold standard for assessing organ damage is ultrasound,
Okay.
which can give us a clear picture of what's happening internally.
Right.
And this is a big
But ultrasound equipment is expensive.
and often inaccessible in the remote communities most affected by schistosomiasis.
So we need a different approach.
Something more accurate.
Accessible and affordable.
Right.
Which brings us to the exciting world of metabolomics.
Yeah.
This is where the science gets really interesting.
Yeah.
Imagine a world where we could diagnose diseases
And track their progression with incredible accuracy.
just by analyzing a simple blood or urine sample.
Okay.
That's the potential of metabolomics.
I'll admit that sounds pretty impressive.
But before we get ahead of ourselves,
Can you explain what metabolomics actually is?
of course.
Think about your body as a bustling city.
Okay.
It's constantly producing, breaking down, and using various substances to function.
These substances are called metabolites, and they act like the city's unique chemical fingerprint.
Now, when your body is infected with something like schistosomiasis, this fingerprint changes in a very specific way.
So it's like the parasites are throwing off the city's rhythm.
And we can detect those disruptions by looking at the changes in these metabolites.
Precisely.
And here's the game-changing part.
Okay.
By analyzing these metabolic shifts,
We can potentially develop much more sensitive diagnostic tests for schistosomiasis.
even in those tricky low-burden cases that are currently flying under the radar, we're eavesdropping on the body's conversation with the infection, kind of like,
We're listening in on the whispers of disease,
all right,
catching those subtle changes that traditional methods might miss.
that's incredible.
Yeah.
But how does this translate into real-world benefits?
For people dealing with schistosomiasis,
That's the key question, right?
imagine being able to diagnose schistosomiasis much earlier,
Okay.
even before any symptoms appear.
That's one of the biggest promises of metabolomics. We're talking about catching the disease in its earliest stages.
Potentially preventing irreversible organ damage down the line.
That's huge.
It's like the old adage, an ounce of prevention is worth a pound of cure.
Right.
But this technology could also revolutionize how we monitor the effectiveness of treatment, right?
Absolutely.
Let's say someone is being treated for schistosomiasis with the standard drug,
Okay.
Bosuquantel.
Mm-hmm.
Currently, we often rely on egg counts to see if the treatment worked.
Right.
But as we've discussed, that's not always reliable.
Right. It's like checking to see if the fire's out just by looking for smoke.
You might miss those hidden embers still smoldering beneath the surface.
Exactly.
Metabolomics allows us to go beyond the surface.
And actually see how the body is responding at a molecular level. Mm-hmm. We can track those metabolic changes over time.
And get a much clearer picture of whether the treatment is working
Okay.
and whether any lingering damage needs to be addressed.
So it's about moving towards a more personalized approach to treatment, tailoring it to the individual's needs based on their unique metabolic profile.
Yes.
It's like upgrading from a one-size-fits-all treatment plan to a bespoke suit.
Perfectly tailored for the best possible outcome.
Precisely.
And the exciting thing is this research isn't just theoretical.
We're already seeing promising results in the lab.
Oh, really?
There was a fascinating study from 2004.
One of the earliest to investigate this approach in schistosomiasis.
Tell me more.
They used a technique called nuclear magnetic resonance spectroscopy.
It's a mouthful, I know.
Okay.
But essentially it allows you to identify and measure different metabolites in a sample.
Okay, so what did they find?
They found that mice infected with schistosomiasis had distinct metabolic profiles
Okay.
compared to uninfected mice.
Okay.
And get this, they even identified specific metabolites that could differentiate between different levels of infection severity.
So even in those early days, they were already uncovering those telltale metabolic clues.
Yes.
And the research has only accelerated from there.
Another study, this one in children, found that specific metabolic changes were detectable just 3 months after their first infection.
3 months.
That's incredible.
It is.
It shows just how early these metabolic shifts occur.
And these weren't just any random metabolites.
Right.
They were specifically related to energy and purine metabolism.
Right.
Okay, I have to stop you there for a second.
Purine metabolism.
For those of us who don't speak fluent science,
Right.
could you break that down a bit?
Of course.
Purines are essential building blocks for DNA and RNA.
Okay.
So they play a vital role in cell growth and function.
Okay.
When the body is fighting off an infection like schistosomiasis, it needs more energy.
And purine metabolism plays a key role in that process.
So seeing these changes in purine metabolism early on,
Could be a red flag that the body is battling this parasitic infection.
even if other symptoms aren't obvious yet.
Exactly.
And here's where things get even more interesting.
Okay.
Remember how we talked about the limitations of using egg counts to assess organ damage?
Yes. How could I forget?
Well,
Researchers have found that certain metabolites in the blood
can actually distinguish between individuals with mild and severe liver damage.
even when their egg counts are similar.
Wow, so we're finally starting to peel back those layers.
And get a clear picture of what's happening inside the body.
Right.
It's like we've had this blurry image of schistosomiasis for so long.
And now metabolomics is bringing it into sharp focus.
It's like putting on a new pair of glasses, right?
Yeah.
We can see the intricacies of this disease in a way we never could before.
And the best part is this technology isn't limited to just schistosomiasis, is it?
No, not at all.
Not even close.
It feels like we've stumbled onto a whole new way.
Of understanding and treating parasitic diseases in general.
Absolutely.
Think about malaria, leishmaniasis, hookworm. These parasitic infections all interact with the body's metabolism.
Right.
If we can crack the code, so to speak.
And learn to read those subtle metabolic shifts.
Yeah.
We could revolutionize how we diagnose, treat, and even prevent these diseases.
On a global scale.
It's like having a superpower.
Right.
The ability to decode the language of disease at a molecular level.
It really is.
But let's bring it back down to earth for a moment.
Okay.
You mentioned earlier that there are still hurdles to overcome.
Before metabolomics becomes a mainstream tool
Sure.
in the fight against schistosomiasis, what are some of those challenges?
Well, one of the biggest is
translating these promising findings from the lab to real-world settings.
right.
In the lab, we can control for a lot of variables.
Yeah.
But in the real world,
Things get a lot messier. People's diets, their environments, their genetic backgrounds, other medications they might be taking.
all of these factors can influence their metabolic profiles.
So it's like trying to solve a jigsaw puzzle,
Right.
where some of the pieces might belong to a completely different picture.
Exactly.
How do we tease apart those confounding factors
And make sure we're focusing on the metabolic changes.
that are truly specific to schistosomiasis?
That is the million-dollar question.
We need robust study designs,
Correct.
larger sample sizes, and sophisticated data analysis techniques,
To filter out that background noise.
and pinpoint the signals that matter most.
And I imagine access to technology and resources plays a role as well.
It certainly does.
Absolutely.
Right.
Right now, Metabolomics relies on advanced and expensive equipment.
That isn't readily available in many parts of the world where schistosomiasis is endemic.
So it's like having this incredible map that could lead us to a treasure. But we need to make sure everyone has the tools and resources.
To actually follow it.
You got it.
A perfect analogy.
Part of the challenge is finding ways to simplify these technologies.
Right.
Make them more portable and more affordable.
So they can be deployed in even the most remote settings.
Like creating a schistosomiasis detecting device straight out of Star Trek.
There you go.
Something portable, user-friendly.
And accessible to healthcare workers on the front lines.
Wouldn't that be something?
And it's not as far-fetched as it sounds.
Really?
There are already researchers working on developing point-of-care diagnostic tools.
That's based on metabolomics.
Wow.
Which could revolutionize how we diagnose and manage schistosomiasis in the field.
So instead of having to send samples to a lab
Right.
and wait days, weeks, or even months for results.
You could get a diagnosis on the spot.
Exactly. And imagine the impact that could have.
Yeah.
Prompt treatment, targeted interventions.
And ultimately, a brighter future for millions affected by this debilitating disease.
This has been an incredible deep dive, I think.
It has.
We've gone from microscopic parasites to cutting-edge science and the future of global health,
Yeah.
all in under an hour.
We've covered a lot of ground.
We have, and we've only just scratched the surface of what metabolomics could achieve.
Just the beginning.
Right. Which brings us to our final thought for today.
If our metabolomes, those unique chemical fingerprints,
Right.
hold so much information about our health,
Not just about schistosomiasis.
but about countless other conditions, what if we could harness that knowledge for more than just diagnosis?
Yeah.
What if we could use metabolomics to develop personalized preventative strategies, identify individuals at risk even before they show symptoms?
And ultimately create a world where diseases like schistosomiasis are a thing of the past.
I like the way you think.
It's a future worth striving for.
Indeed it is.
Yeah.